you very much. Thanks for the opportunity to talk to you all today. And I'm going to present ISS as a research test bed with all these constraints in mind from a little bit of a different perspective, and that is from the scientist's perspective, from the investigator who gets to think about how to utilize this environment, us taking this knowledge and applying it to work that on Mars surface eventually. Am I not using this, or is this?
No, you can do the call out.
Or I can do it myself. I like doing things myself. Okay. So ISS as a research platform. Well, what makes this so unique? And I think it's been spoken of already, and we all know this, that it's the sustained conditions of reduced gravity, of microgravity, that make this a laboratory like none other that currently exist. There are definitely ways of reproducing some of the aspects of microgravity using simulations with drop towers or with random positioning devices for cell work, or with bioreactors, with various kinds of parabolic flight. But the bottom line is, in order to get sustained experimental opportunities in microgravity, this is it. This becomes important for a variety of reasons, and several of those reasons surround on real nuts and bolts kind of things like hardware development. If I'm going to do an experiment, I have to have the hardware capabilities to be able to support the experiment and to give me the kinds of results that I'm going to need to have. So that might be modification of existing hardware, or that might be the whole new design of hardware. These are sorts of things that can be modeled using the ISS environment with an eye towards how these capabilities might work on a trip to Mars. In addition, we have to be able to define the kind of experimental paradigm that we're talking about here, and whether that's a biological or a chemical or a physical experiment or materials science. Each of these represents their own set of requirements that are important in order to get the kind of data one would like to see. There becomes the question about can we make the experiment run autonomously, and that's going to factor back to the hardware. What kind of crew interface are we going to have? How much crew time are we going to need to be able to impact the experiment to get the kind of results that we're looking for? And then finally, and this is a really big question that impacts all kinds of experimentation, is what sorts of sampling, analysis, and storage requirements are we going to be looking at? Right now on ISS, these are the discussions that we're having, and that's why this is so important to think of this in the context of leaving low Earth orbit and how we can take our experiences that we gain from understanding the difficulties we have to overcome to do good experimentation on ISS to go in farther. And we can't do this without partners, as we've said before. And I'm going to present some of our work with the ISS National Lab Pathfinder missions as sort of a model for how, at least in my mind, we can apply this platform to work going beyond low Earth orbit and onto Mars. This cannot be done without each of these partners. The work that I'll be talking about today started out as a fundamental science finding from a group, and then we had the opportunity to ask a question: if we could understand that some of what's going on with that finding, can we use that to develop a real way to treat health on Earth? And can we use microgravity to help us answer some really important questions? And so the academic partner is very strong here, and the work that I'll be talking about today is Dr. Tim Hammond is the principal investigator at Duke, who oversees the nuts and bolts of the bench time. And believe me, anybody who's done this work knows that there's a lot of bench time on the ground required in order to make it a good flight study. We have a strong industry partner, and the partner actually has been funding us all along for this. And this is Astrogenetics, which is a privately held subsidiary of Astrotech, and a lot of you will know that as SpaceHab. And they came to the table with funding so that we were able to do the experiments that then led to the development of the formation of this little subsidiary. And finally, we wouldn't be able to do any of this without government, without the flight opportunities that have allowed us to kind of model how we're going to be doing this on National Lab and how other groups can use this information to similarly develop their platforms, whether that be industry, academia, or other governmental entities. It takes all three, and you can't do it with just two. It's tough. So again, I'm going to present what we've done sort of as a model in your minds, be thinking how this could be applied to work beyond low Earth orbit, because everything that I'm talking about today has been done on shuttle. And we fully anticipate that this work will be continued in National Laboratory. So basically, we utilize a basic science finding that centers on the fact that bacteria change when they get exposed to microgravity. And so if that happens, we thought we could potentially be able to apply this knowledge to understand developing new treatments to treat infectious disease on Earth. We know this is a very costly problem, and it results in very high morbidity and mortality. And we want new therapeutics not only to spread stop the spread of disease, but also to be able to circumvent the antibiotic resistance that is happening with lots of organisms these days. So our approach in developing these National Lab Pathfinder vaccines was to take advantage of the changes that happen in bacteria that are uncovered by microgravity. And so this is where the platform for new discovery comes in, because these are not changes that would normally be experienced here on Earth. We can drive them to be experienced in ground-based lab. We can make the bacteria do things, but the uncovering of that is something that we really can't replicate unless we have true microgravity. Our approach has been to look at changes in gene expression and to use gene targets as a foundation for the development of new types of therapeutics. And so we actually did our first experiment, and we initiated this group and started moving out with the work, starting with STS in March of 2008. So this is a little bit of a busy slide, but as I mentioned, when you take bacteria and put them in space, they change. That's the end point. I got a little bit moved there. I don't know why, but anyway, yeah, see, it's happening already. You're exactly right, Sam. But they change in their ability to infect a host. They may change in their antibiotic resistance. They change in their growth patterns. They change in many ways that we're just beginning to understand. Again, so we use this as a means of developing some engineered bacteria, and so we made some deletions. And we developed a flight study where we used a little host, and this is a little worm, a little nematode, C. elegans, and it actually replicates, fortunately or unfortunately, a lot of aspects of the human immune system, believe it or not. So to think that we're that related to worms is a little bit scary, but in our case it has worked really well. So we then developed a flight testing paradigm using hardware that's provided by BioServe, and we've repeatedly done these studies now, and we actually are at the point where we're in the process of rounding up the data and putting it together for submission of an investigational new drug package, which is a huge initiative to put that forward to the FDA, with the hopes of someone picking this up and actually going on to develop new therapies and commercial vaccines. So basically we leveraged a lot. We leveraged the ground-based work to identify the candidate genes that we were interested in examining. We got the best folks, and our model organism in this work was Salmonella. We got some really great Salmonella people to help us go in and make intelligent choices of what genes are you going to be knocking out, and then looking at what happens in the space environment. We leveraged ground-based assays to optimize the host microbe interactions, so we could get the assay time down, and we could get the endpoints driven to the places that we needed to see outcome. And we used existing flight hardware that we actually build each time. If any of you have used the BioServe hardware, it's the GAP. So it's basically like a pineapple can with 8 test tubes, and you can build each of the test tubes yourself with inserts. And then we went on to validate this and develop an in-flight host pathogen assay. So how does this all work? Well, we have these strains of bacteria, and in each of the parts of the little test tube here is a different component. The first part will be the bacteria. In the beginning studies, this was salmonella. The next part will be the little C. elegans worms. And then this is all loaded up into the can. The last part here is fixative. So in order to start the experiment, the crew will take a crank and actually depress these first two areas and mix the bacteria and the little worms together, and they'll hang out for the period of the assay. And then after a set period, and we've got most of these assays down to about a 48-hour period, the crew does one more crank, and the experiment is stopped, everything's frozen in time, and we can take that and look at it and see what we got from the experiment. So how does this work? Well, they're brought together, they're launched separately, the bacteria and the worms, they're brought together in space. If the bacteria infect and kill the little worm host, the worms are eliminated, and so the bacteria will continue to grow. And so that's reported as increased optical density, or a really opaque-looking solution. If the bacteria do not infect and kill the little worm host, they're eliminated because the worms eat them, because that's what worms do, is they eat these little bacteria. And they're not there, and so you don't see the increase in optical density that's reported at the end of the experiment. Let me go back one. So why is this important? And I was talking about knocking out genes to get rid of virulence, and bacteria, when they go into space, become more virulent. Well, what does that mean? Well, we reasoned that if we could figure out the genes that were responsible for the increased virulence, and if we could take them away one by one, and then identify that if we took away the gene, then we took away that increased virulence effect, that might become a new target that we could use to put forward for a new therapeutic, for a vaccine or for some other kind of antimicrobial therapeutic. And this is the hallmark of creating what's called an attenuated vaccine in science jargon, and that is a vaccine that will not make the host sick, but will allow the host, when they get immunized with it, to be able to defend against future invasions of that bacteria. And so that is exactly the course that we set out to achieve using this line of investigation. And so this is from our second flight study, and we use a wild-type worm, so the worm hasn't been modified. And we do something kind of tricky, and we actually alter the worm in one of the areas, a receptor that the bacteria must kind of latch onto so it gets ingested by the worm. So we're actually looking at responses of the bacteria as well as by the host. So it's kind of cool, we thought. And we've gotten some really, it's turned out very well. We've gotten some good data with this. And in this experiment, when we delete this gene number 4, we don't really see any change in virulence relative to the wild type. When we delete number 3, no change. But when we delete number 1 or number 2, we saw that the increased virulence effect went away. And so this is salmonella, and we're actually pursuing these 2 specific genes as to engineer a double knockout, and that will become part of our IND package. So what have we been able to do? Well, basically, in 3 years, we've manifested and accomplished 11 payloads. The first 3 of them were dedicated to understanding salmonella and identifying target genes. And then we asked the question, well, if salmonella does this, what about other organisms? And so we did a couple of survey flights with other medically relevant bacteria, and we were able to use salmonella as our positive control. And in science, the big deal is using your positive control so you know what you're going to get, so that you can understand when you fly your unknowns, the new bacteria, you can understand the responses that you're seeing. And then one of these showed particular promise, methicillin-resistant Staph aureus, or MRSA, and everybody's heard about MRSA and the problems that causes in the hospital, as well as now in the community, in gyms and schools, and it's a really huge problem. And we, with 128, we started our MRSA studies, and we've actually identified a couple of genes for targets in this organism as well. So we've flown 8 medically relevant bacteria and 1 important yeast, Candida albicans. And I get this question all the time, this is nice, it's interesting science, I can see where this is going, but how is this going to change anything on Earth? And remembering again that we're patterning this work to be a model for work that can be done in National Laboratory, with the mission, in fact, of using the information that's acquired in that environment to create benefit on Earth. And so those of us that are familiar with R&D pipelines for drugs and other kinds of applications, the pipeline for a single agent can take years and years of work. It'll allow you to identify a lot of good candidates that have pharmaceutical applications, but at the end of the period, when you actually get into the point of testing these targets, it's typical that most of them will not be suitable for continued development. So the resources expended getting to that point, if there could be a way to fast track the identification of promising targets and to then fast track those on if they in fact are promising, or to terminate them and not spend any more resources, time, money, and effort on developing them, would be of major economic impact. And so we do then postulate that using space, we could eliminate years from the R&D pipeline to allow these kinds of fast-tracking activities. And I don't really, this is a YouTube, you can go on and find it online, but you see John Phillips working with the experiment, and you'll see him cranking it here to initiate it, and then he gives one final kind of a turnover of the gap, and the experiment is on its way. So just to summarize our progress, it's important to note that at the foundation of all of this, the findings that have been made in space are the product of good fundamental research. And it's that research finding that facilitated the development of this venture that has been supporting our work going forward. Since 2011, we've manifested 11 payloads, all with industry support. And we think this is a proof of concept for operations on ISS National Lab. And I would then take this one step farther and say that the strength of ISS as a research platform for discovery and autonomous operations can be applied to missions beyond low Earth orbit.